All right, let's jump right into this explainer. We're going on a seriously mind-bending journey today. We're starting way back at the chaotic, literally boiling scale of early Earth and zooming all the way down to the invisible, microscopic boundaries that are keeping you and me alive right at this exact second. We go through our whole day completely ignoring the tiny machinery running the show. But by the end of this, I promise you're going to look at the fundamental units of life in a totally new, crystal clear way. So, where did it all start? To understand how we're built, we have to rewind. Picture Earth, about 3.5 billion years ago. It's intense. Now, scientists generally agree that life kicked off in water, right? But we're not talking about some calm, gentle ocean breeze. We're talking extreme pools of water with wildly fluctuating conditions. Which brings up a really fascinating question. In an environment absolutely blasting with extreme heat and nasty radiation, how on earth did fragile, early biological stuff even survive long enough to form a functioning cell? Well, here's where it gets incredibly cool. We actually have environments today that mirror that ancient chaos. Take the hot springs of Puganugu Valley over in Ladakh. Even in a freezing climate, these pools are bubbling away, sitting right near the boiling point. Researchers from the Birbal Sahni Institute of Paleosciences went out there specifically because it's a dead ringer for early Earth. And what they found was wild. They saw rapid calcium carbonate deposits forming right around these boiling pools. These minerals likely acted like a microscopic shield, right? They protected those early organic molecules from the harsh conditions, giving them the chance to form the very first protective membrane. That is the ultimate, definitive barrier that actually makes a cell, well, a cell. Fast forward a few billion years, and these cellular structures, they're everywhere. They are the fundamental building blocks of literally all living things, from a single bacterium to you, sitting there with millions of them making up your body. So why did it take humanity thousands of years to even notice them? It basically boils down to a hard physical limit of our own biology. That number you're seeing, 0.1 millimeters, that's our roadblock. That is the absolute limit of what the human eye can handle. Scientists call this the limit of resolution. Basically, if two tiny points are closer together than 0.1 millimeters, our eyes physically can't tell them apart anymore. They just blur into one single dot, even if you're holding them at a normal reading distance. And since almost every cell on the planet is way smaller than that threshold, they were totally invisible to us. We were literally blind to the very things we're made of. But humans are tool builders, right? We eventually smashed right through that visual limit. Back in 1665, a brilliant guy named Robert Hooke designed his own microscope. It could magnify things maybe 200 to 300 times. So he puts a super thin slice of cork under the lens, maybe a leftover from a good bottle of wine, who knows, and he sees something totally revolutionary. He sees these little box-like compartments, and he famously calls them cells. This amazing invention, the light microscope, used a combo of objective and eyepiece lenses to finally snap the invisible world right into focus. And the crazy part is, you don't even need a supercomputer to measure these microscopic things. It's actually a pretty clever trick. You just take the total diameter of your microscope's field of view, let's say it's 5,000 micrometers, and you count how many cells fit perfectly across that line. If you count 25 onion cells, you just do some quick division, and boom, you know each one is exactly 200 micrometers wide. It's just a brilliantly simple way to map out an entirely invisible world. Of course, light microscopes are fantastic, but eventually, scientists wanted to go deeper. So today, we actually bypass light completely. Enter the electron microscope. Instead of bouncing light, it fires a literal beam of electrons to create massively magnified images. This lets us zoom all the way down to the nanometer scale. To put that in perspective, a nanometer is 1/1,000,000th of a meter. With that kind of jaw-dropping clarity, we can finally map out the insanely complex, fine details hiding inside the cell. And when we use that tech to look at the very edge of the cell, we find this absolute masterpiece of biology, the fluid mosaic model. This is the universal boundary of the cell membrane, and it's shockingly thin. We're talking only 7 to 10 nanometers. It's built out of a lipid bilayer, which is basically special fat molecules. They have water-attracting heads that face the outside world and water-repelling tails that hide on the inside. But here's the kicker. It is not a rigid brick wall. It's actually fluid. The molecules are constantly swimming sideways, flipping and spinning around. And floating inside this shifting sea of fats are proteins. They look like tiles in a mosaic, and they act as these highly selective gatekeepers, deciding exactly what is allowed in or out. So why does having a fluid, highly selective gatekeeper matter so much? Well, let's put it through a stress test. What happens when the cell's environment gets thrown out of whack? We have to look at extreme scenarios involving solutes, like salt or sugar, dissolved in the water outside the cell. In a hypotonic environment, there's less of that solute outside the cell than inside. Think of it like dunking a cell in pure fresh water. On the flip side, a hypertonic environment means the solute concentration outside is way higher than inside. Imagine a heavy, syrupy salt or sugar solution. Let's look at what that actually does to living tissue using something as simple as a potato. Over on the left side, we put a potato piece in that hypotonic, plain water environment. Because of osmosis, water aggressively rushes into the potato cells, and the whole piece actually swells up. But watch what happens on the right. When we drop a piece into a hypertonic, 20% salt solution, osmosis throws everything in reverse. The water rushes out of the cells trying to balance things, and that massive water loss causes the entire piece of potato to severely shrink and shrivel up. Now, this creates a major life-or-death problem in nature. Think about it. If you're a dog or a bird and your environment gets way too salty or dry, you just get up and move, right? But a plant is literally rooted in the dirt. It has to just sit there and take whatever extreme weather comes its way. So how on earth do plant cells survive these wild environmental swings without just constantly swelling up and bursting or shrinking into nothing like those potato cells? The answer is a seriously impressive evolutionary upgrade, the cell wall. This is a rigid, tough layer of armor found on the outside of the cell membrane, but only in things like plants, fungi, and bacteria. It's an absolute game changer for survival. If a plant cell gets flooded with rain and starts swelling, that cell wall acts like a literal cage, physically stopping it from bursting open. And during a severe drought, when the plant is losing water, the squishy inner membrane might shrink away, but that stiff cell wall, it holds its ground. It maintains the shape and keeps the whole plant from just flopping over. You can actually see the sheer structural power of this armor when you put plant and animal cells side by side. Look at the plant cells from an onion peel on the left. See how perfectly boxy they are? They're stacked neatly together like rigid little bricks. That is the cell wall doing its job. Now pan over to the human cheek cells on the right. Animal cells don't have that wall, so they are totally irregular, kind of squishy and loosely arranged. They rely completely on that thin, fluid mosaic membrane to keep everything contained. It really leaves you with a profound sense of awe, doesn't it? We kicked off this whole explainer looking at the violent, boiling hot springs of ancient Earth, seeing how simple minerals sheltered the very first sparks of biology. And now, we realize those ancient survival mechanics are literally inside us. Those exact same microscopic, selectively permeable, shifting boundaries that define the first life forms are silently, relentlessly working across millions of your cells right now, just keeping you alive. It makes you wonder, what other invisible miracles are happening right beneath our limit of resolution?